Hi, my name is Ashley Walker. I'm an assistant professor in the Department of Human Physiology at the University of Oregon. One of the hallmark features of advancing age is an increase in stiffness of the large elastic arteries. Recent studies in humans have found correlations between large artery stiffness and cerebrovascular dysfunction, cognitive impairment, and an increased risk for Alzheimer's disease. However, the mechanisms underlying this link between large artery stiffness and poor outcomes in the brain are unclear. In these studies, we examined the effects of large artery stiffness on the cerebral artery response to vasoconstrictors. To examine the causative nature of this relation, we used a transgenic mouse model of large artery stiffness, the elastin heterozygote mouse. To perform these studies, we used isolated and pressurized middle cerebral arteries. We found that the vasoconstriction response to angiotensin II was greater in cerebral arteries from elastin heterozygote mice compared with wild-type mice. By using losartan, we demonstrated that this greater vasoconstriction was mediated by the angiotensin II type 1 receptor. To summarize, in these studies, we found that in a mouse model of greater large artery stiffness, there was a greater vasoconstrictor response to angiotensin II in the cerebral arteries. We also examined the dose responses to potassium chloride, norepinephrine, and endothelin-1 and found no difference between cerebral arteries from elastin heterozygote and wild-type mice. In parallel with these experiments in cerebral arteries, we also examined the response to vasoconstrictors in the gastrocnemius feed arteries. In this case, we found no difference between groups of elastin genotypes on any of the vasoconstrictor responses. These findings align with a previous study by our group that demonstrated that the elastin heterozygote mouse has impaired endothelial function in cerebral arteries, but not skeletal muscle feed arteries. These findings also align with the human literature, where it is found that the cerebral vasculature is one of the most susceptible circulations to the effects of large artery stiffness. Overall, these results indicate that increased responsiveness to angiotensin II may be one mechanism linking age-related increases in large artery stiffness to cerebrovascular impairments.